Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1. To help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2. To build a supportive community where we can exchange the most valuable experience-based research advice. 3. To share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Okay, so think about this. You've got, what, 2 meters of DNA packed into this tiny, tiny cell nucleus. You'd probably picture it as just a total tangled mess, right?
That's the common image, definitely.
But what if we told you it actually folds up in this super organized way, completely unknotted, like elegant biological origami that allows everything to work properly?
Exactly. And today we're diving into how scientists figure that out. Back in 2009, a groundbreaking method called Hi-C revealed this amazing secret, something called the fractal globule.
Welcome to the deep dive. Yes, today we're exploring how researchers finally mapped the incredible 3D architecture of the entire human genome, uncovering these fundamental folding rules.
We've got the original paper right here, and we'll unpack just how revolutionary this Hi-C technique really was.
Our mission today: break down the ingenious method, look at the really surprising discoveries, and understand the lasting impact. Because this completely changed how we think about our DNA's organization.
Yeah. Before 2009, how DNA fit into the nucleus was, well, honestly, one of biology's biggest puzzles. We weren't totally in the dark, mind you.
Right. We had some clues.
We did. Microscopy showed chromosomes seemed to have their own sort of zones or territories. They weren't just mixing like spaghetti.
Okay.
And earlier methods, things like 3C, could check if maybe a few specific DNA locations were close to each other, but it was super limited. Like knowing only 2 or 3 street addresses in a whole giant city.
So hints of connections, but no overall map. No real geography of the genome's 3D structure. That sounds like a massive hurdle.
It really was. I mean, the core problem. How do you pack 2 meters of DNA into a nucleus that's only, what, a few micrometers across?
And crucially, does it fold randomly? Or are there rules, universal principles?
Exactly. And this wasn't just, a matter of satisfying scientific curiosity. That 3D arrangement is absolutely critical for how cells function.
How so?
Well, think about gene regulation. Turning genes on or off. Often a control element, an enhancer, might be really far away from a gene in the linear sequence.
Right.
But in 3D space, it needs to loop over and physically touch or get very close to that gene to activate it. Spatial proximity is key.
And things like DNA replication, too.
Yes, and genome stability, making sure everything is organized correctly. Our models for all these vital processes were built on really sparse data, just these few known contact points. The field desperately needed a way to see all the contacts, genome-wide.
So a two-part challenge, then. First, invent a completely new technique, something comprehensive and unbiased to capture all those interactions at once.
Right.
And second, take that mountain of data and figure out the underlying folding principles, so sort of bridging the gap between molecular biology and, like, polymer physics.
Precisely. It needed that interdisciplinary thinking, and that's where Hi-C came in. It was such an ingenious design.
An evolution of the earlier methods, you said?
Yes, a clever generalization of 3C, really, designed to capture these snapshots, let's say, of all the points where different DNA segments are physically touching inside the nucleus.
Okay, let's walk through it. How do they actually do that? It sounds incredibly complex.
The steps are actually quite elegant when you break them down. First, they treat the cells with formaldehyde.
Okay, the preserving agent.
Exactly. It acts like a molecular superglue. It creates tiny chemical crosslinks between proteins and indirectly between DNA strands that are right next to each other in 3D space, basically freezing those interactions.
Got it. So you've locked in the 3D structure as it was in the living cell. What's next?
Then they use restriction enzymes, these are like molecular scissors, to cut the chromatin, the DNA protein complex, into millions of fragments.
Okay, chopping it up.
But here's a key trick. While cutting, they fill in the sticky ends that are created, and they incorporate a special nucleotide that has a biotin tag attached, a tiny chemical flag.
Biotin, like the vitamin.
The very same.
Oh.
It acts as a handle they can grab onto later. So now, the ends of the fragments that were close enough to be cross-linked have this biotin marker right near the junction point.
And then comes the really innovative part, the dilute ligation. Correct. That seems critical.
Absolutely crucial. This is the core of Hi-C. They take these millions of cut fragments now floating around, and they put them in a very, very dilute solution.
Why dilute?
Because in dilute conditions, a DNA fragment is much more likely to bump into and join, or ligate, with the fragment it was originally cross-linked to the one right next to it in 3D space, rather than some random fragment from across the nucleus.
Ah, okay. So the dilution forces nearby fragments to rejoin.
Precisely. This creates new hybrid DNA molecules, these chimeric molecules, where two segments that might have been far apart on the linear chromosome are now joined together. And crucially, that junction point is marked with biotin.
So each of these chimeric molecules is like a little record of a 3D interaction that happened in the cell.
Yeah.
But how do you find those specific molecules among all the other DNA?
Right, you got the whole genome chopped up. So next, they shear all the DNA into much smaller pieces. And then they use those biotin tags. They use beads coated with streptavidin, a protein that binds very tightly to biotin.
Like molecular fishing.
Exactly. They fish out only the fragments containing biotin. And since biotin is only at those ligation junctions, they are specifically isolating the pieces of DNA that represent the original 3D contact points.
Brilliant. So you've purified the evidence of proximity.
Yes. And then the final step is massively parallel sequencing. They feed all these isolated junction fragments into high-throughput sequencers.
Which read the DNA sequence of both ends of each fragment.
Correct. So if you sequence both ends of one of these chimeric fragments, you find out which two parts of the genome were originally close together in 3D space. You do this millions of times.
And you get a massive genome-wide map of all the contacts, an interaction matrix.
That's it. And what's really important is that they didn't just invent this. They tested it rigorously. They generated something like 100 million sequence reads from human cells, mapping hundreds of thousands of unique contacts.
Wow.
They visualized this data as heat maps, these contact matrices you see in the paper. And critically, they repeated the whole experiment using a different restriction enzyme, a different molecular scissor.
And got the same patterns.
Almost identical results. That reproducibility was key. It showed the patterns they were seeing were real features of genome organization, not just some artifact of the specific enzyme they used.
That's crucial for confidence. Okay, so with this reliable, powerful tool, what did they find? What did this new map of the nuclear landscape look like?
Well, first off, the data immediately confirmed something suspected for a long time. Chromosome territories.
Meaning each chromosome kind of sticks to its own area.
Pretty much. If you look at interactions within a single chromosome, the probability that 2 points interact obviously goes down as they get further apart along the DNA strand. Does that make sense?
Like points on a string?
Right. But what was striking was that even points that are very far apart on the same chromosome, say 200 million bases apart, are still much more likely to interact with each other than they are with points on a different chromosome.
Ah, okay. So chromosomes mostly keep to themselves. They have their own neighborhoods.
That's the idea. Each chromosome seems to occupy a relatively distinct territory within the nucleus.
But do they ever interact? Do these neighborhoods connect? What about contacts between different chromosomes?
Ah, yes, the interchromosomal contacts. That's where things got really interesting and revealed some, let's say, social preferences.
Chromosome cliques.
You could say that. They found that small chromosomes that are packed with genes, the gene-rich ones like chromosomes 16, 17, 19, 20, 21, and 22, they tend to interact preferentially with each other. They often cluster together, typically towards the interior of the nucleus.
So the busy, active chromosomes hang out together. The social butterflies of the genome.
That's a fun way to think about it. And in contrast, they looked at chromosome 18. It's also small, but it's relatively gene-poor.
The quiet one.
Yeah, and it tended to be located more towards the edge of the nucleus, the nuclear periphery, and it didn't interact nearly as much with that group of gene-rich chromosomes. So Hi-C started revealing these preferential associations.
That's fascinating detail we just didn't have before. But there was even more structure, right? As they zoomed in, they saw this plaid pattern.
Yes, that was really remarkable.
Yeah.
When they looked at the interaction map within a single chromosome, it wasn't smooth. There were these large blocks, megabases in size, where interactions were either much higher or much lower than expected. It created this distinct checkerboard or plaid pattern in the heat map.
Suggesting another layer of organization within the territories.
Exactly. And analyzing this plaid pattern led to a major discovery. The entire genome, essentially, can be divided into two large-scale spatial compartments. They just call them A and B.
A and B compartments. Yeah. Okay, how distinct are these?
Very distinct.
Yeah.
They found that DNA segments located within the same type of compartment, so A with A or B with B, interacted frequently with each other, even if they were far apart linearly. But interactions between segments in A and segments in B were surprisingly rare. It's as if the nucleus is partitioned into these two major environments that don't readily mix.
Like oil and water, almost.
That's a good analogy. And they even confirmed this visually using microscopy with a technique called 3D FIH.
Right. Lighting up specific DNA spots?
Yes. They showed, for example, that two loci, two spots on the DNA, that Hi-C said were in compartment A, were consistently closer together in 3D space than one of them was to a locus from compartment B, even if that B locus was linearly right between them on the chromosome.
Wow, so 3D proximity really doesn't follow the linear map at this scale. It follows the AB compartment assignment.
Precisely. It showed the compartments were real spatial entities.
So what are these A and B compartments functionally? What do they represent?
They found really strong correlations. Compartment A regions strongly correlated with areas rich in genes, especially actively transcribed genes. They also correlated with open chromatin DNA that's accessible to the cellular machinery, and with chemical marks on histone proteins that signal active genes.
So A is the active, busy compartment, the open for business parts of the genome.
You got it. And compartment B, that tended to be the opposite. More gene-poor regions, less transcription, more densely packed, closed, or inaccessible chromatin associated with repressive histone marks, the quieter parts of the genome.
Okay, active A, inactive B. Makes sense. But is this static? Or can it change?
That's another key finding. It's dynamic. They compared Hi-C maps from different cell types, like a normal lymphoblastoid cell line and an erythroleukemia cell line.
Yeah.
While the overall AB patterns were similar, many specific regions actually switched compartments between the cell types. A region that was B inactive in one cell type might become A active in the other.
And did that switch correlate with gene activity?
Perfectly. The switch in compartment status correlated precisely with changes in gene expression and chromatin accessibility for that region. It showed these compartments aren't fixed. They reflect the dynamic functional state of the genome in a particular cell type.
That's huge. The 3D structure is actively reflecting and maybe even influencing cell function and identity. Okay, so we have territories, AB compartments, but then there's the really mind-bending part, the fractal globule.
Ah, yes. This came from looking even closer within the compartments. They analyzed how the probability of contact falls off with increasing linear distance along the DNA, specifically in the range of about 500,000 bases up to 7 million bases.
And they found a specific mathematical relationship, a scaling law.
Exactly. They observed a very specific power law scaling. Contact probability decreased as distance to the power of -1.
Okay, S to the -1. Why is that significant? What was the old idea?
Before Hi-C, the dominant model for how chromosomes might fold locally was something called the equilibrium globule. You can picture it like a tangled ball of yarn or spaghetti crammed into a bowl. It's compact, yes, but simulations showed it would be incredibly knotted and entangled.
Which sounds bad for a cell needing to access its DNA.
A complete nightmare. Imagine trying to quickly find and pull out one specific piece of yarn from a tightly knotted ball without discarding everything else. Plus, that equilibrium globule model predicted a different scaling law, S32, which just didn't match the Hi-C data.
So the data ruled out the simple tangled ball model.
Correct. And it pointed towards this alternative, maybe less intuitive model, the fractal globule.
Fractal globule. Okay, break that down for us. It sounds complex.
It's actually quite beautiful. Imagine folding the DNA, not randomly, but hierarchically. Small loops fold into slightly larger loops, which fold into even larger loops. You get these globules of globules.
Okay, a hierarchy of loops.
The absolute key property of this fractal globule structure is that it's unknotted. It achieves high density, high compaction without creating knots or entanglements.
Unknotted. How is that even possible when it's so crammed?
It's a property of this specific type of fractal folding. Think of it like carefully crumpling paper versus just randomly stuffing it in your pocket. One way avoids hard knots. And this unknotted structure is perfect for biology. Because it's compact, yet highly accessible, regions of the genome that are close together linearly tend to stay close together in 3D space, forming local domains. This makes it incredibly easy for the cell to, say, spool out a specific gene or region for transcription or replication, and then fold it back up without having to untangle the entire chromosome.
Ah, so it balances density with accessibility, the best of both worlds.
Exactly. Maximal density, minimal entanglement, easy, local unfolding. It's an incredibly elegant solution to the packing problem.
And they confirmed this with simulations, too.
They did. They ran computer simulations creating both equilibrium globules and fractal globules. The fractal globule simulations perfectly reproduced that S1 scaling law they saw in the real Hi-C data. The equilibrium globule simulations produced the S32 scaling. It was strong evidence supporting the fractal globule model for DNA folding at that megabase scale.
Amazing. So let's put it all together. What's the picture of genome architecture Hi-C revealed?
It's this fantastic multi-scale organization. At the very largest scale, you have the genome partitioned into the active A and inactive B compartments.
Okay.
Then, you have individual chromosomes mostly occupying their own territories within the nucleus.
Got it.
Within those territories, each chromosome kind of snakes back and forth, weaving its segments between the A and B compartment environments.
Right, reflecting its local activity state.
And then, at the scale of roughly 500,000 to several million base pairs, the DNA fiber itself is folded up into these unknotted, accessible fractal globules. It's just an architectural masterpiece.
It really is. And thinking about the innovation here, this wasn't just a technical tweak. It feels like it was driven by a really deep question.
Absolutely. There was this fundamental curiosity about how chromosomes self-organize,
Yeah.
but also a critical need. How could we understand gene regulation or the effects of mutations in non-coding DNA if we didn't know which pieces were actually interacting in 3D space? The field knew it needed a whole genome 3C to move beyond anecdotal evidence.
And the leap in scale Hi-C provided was just enormous.
Mind-boggling, really. Previous methods like 3C, 4C, 5C could maybe look at hundreds, perhaps thousands of interactions if you really pushed it. Hi-C, by swapping targeted primers for this bioencapture and deep sequencing approach, blew the search space wide open, from maybe 102 or 103 interactions to potentially 108 pairs or more.
Wow, that's orders of magnitude.
It allowed them to get kilobase to megabase resolution across the entire genome, limited basically only by how deep you could sequence, how much data you could afford to generate.
And this breakthrough wasn't the work of just one lab, was it? It sounds like a major collaborative effort.
Definitely. It was a perfect example of interdisciplinary science. You had Jobdecker's lab with the expertise in chromatin conformation capture methods like 3C, really optimizing the wet lab assay.
Okay.
Then you had Eric Lander's group at the Broad Institute, providing the, at the time, almost unbelievable sequencing power and the computational muscle to handle that massive amount of data.
Essential for genome-wide analysis.
And crucially, you had Leonid Merney's group at MIT, bringing the polymer physics perspective, the theoretical modeling that was essential for interpreting the data and proposing the fractal globule model. That combination was incredibly powerful.
And key individuals driving it forward.
For sure. People like Erez Lieberman-Aden, who made the conceptual leap connecting the data to polymer models and worked on the biotin strategy. Ninky van Berkom, who meticulously developed and performed the wet lab experiments. Jobdecker, guiding the methodology. Eric Lander, enabling the scale. A real team effort.
And they took a big risk, didn't they, going straight for the human genome?
Yeah, that was bold. Many groups might have started with yeast or bacteria, something simpler, but they went right for the complexity of the human genome, which really paid off in terms of impact.
So let's talk about that impact. How did Hi-C fundamentally change things, in terms of theory first?
Well, as we discussed, it provided the first strong experimental evidence for the fractal globule model of polymer folding in a biological context. That was huge for physics and biology. It also, importantly, paved the way for discovering TADS topologically associating domains a few years later. These are smaller, highly self-interacting domains that seem to be fundamental building blocks within the compartments.
And technologically.
It just opened the floodgates. Hi-C launched a whole new field of genome-wide chromosome conformation capture. It spawned numerous variations and improvements, things like in situ Hi-C, which is done inside intact nuclei, micro-C, which uses a different enzyme for higher resolution, even single-cell Hi-C to look at variability between individual cells.
So it created a whole toolkit for studying the 4D nucleome, the nucleus in space and time.
Exactly. These methods are now fundamental tools for anyone studying chromatin structure and function.
And how are these Hi-C maps actually used now in research and maybe even clinically?
Oh, indispensably. They're used to interpret the function of non-coding genetic variants found in disease studies. Maybe a variant doesn't change a gene, but it disrupts a crucial 3D loop that controls a gene far away.
Ah, linking structure to disease.
Yes. They help guide CRISPR gene editing experiments if scientists want to, say, create or break a specific DNA loop to test its function. and they're essential for building and validating sophisticated computer simulations of chromatin dynamics.
It's amazing how one technique could have such a ripple effect. Of course, the initial Hi-C had limitations, right? The resolution wasn't super fine-grained, and it averaged signals from millions of cells.
True, the first maps were around 1 megabase resolution, and you lost all the cell-to-cell variability. But that initial conceptual and methodological leap was the critical thing. It completely redefined the questions we could ask about genome architecture.
It really ignited this whole field of 3D genomics over the last decade and a half.
Absolutely. Nearly every modern study looking at large-scale chromatin folding builds on the foundation laid by that 2009 Hi-C paper.
So wrapping up, what started as this quest to just map contacts to see what touches what inside the nucleus
Blossomed into this profound discovery about how our genetic blueprint is so elegantly organized. Not a tangled mess, but this dynamic, accessible, functional structure, the fractal globule within compartments and territory.
It's an amazing story of scientific discovery.
It really is. And this understanding, this shift from tangled spaghetti to fractal origami, it doesn't just solve the packing problem. It fundamentally changes how we need to think about gene regulation, development, and disease.
Which leads to a final thought for you, our listeners. Given what we now understand about this intricate dynamic folding, this fractal architecture, what new possibilities does this open up? Could we potentially treat diseases by targeting or correcting faulty genome folding? And maybe even more broadly, how might we leverage these fundamental principles of DNA organization to engineer biological systems in entirely new ways?